Thank you very much for inviting me to the symposium. I'm very happy to be here today. My name is Walaa Darwiche. I am Research and Development Engineer at the Amiens University Medical Center. I work in the north of France. I work in the cytogenetics and constitutional genetics laboratory. In our laboratory, we focus on the detection of genomic aberrations responsible for human diseases at molecular level and at the chromosomal level. We detect both unbalanced variations such as deletions, duplications, and balanced variations such as insertions, inversions, and translocations. These genomic abnormalities may lead to genetic diseases, infertility, as well as congenital malformations. To identify these abnormalities, we use in our lab several complementary techniques such as karyotyping, fluorescence in situ hybridization, chromosomal microarray analysis, qFPCR for aneuploidy detection, short and long read sequencing, Sanger sequencing, and more recently, optical genome mapping. I will not go into technical details about how optical genome mapping works today, but it's a technique that analyzes ultra-high molecular weight DNA in order to detect translocations and copy number variation. We use in our lab these techniques in various indications, including prenatal testing, infertility, developmental disorders, hereditary cardiac diseases, and rare disease. We know that chromosomal microarray analysis is very efficient for detecting unbalanced chromosomal variations, such as aneuploidies, deletions, and duplications. However, balanced rearrangements, such as translocations and inversions, are not detected by microarray. These abnormalities are usually identified by karyotyping, which has a low resolution and often require targeted FISH analysis for confirmation. In addition, karyotype requires dividing cells, is labor intensive, and its interpretation is subjective and highly dependent on expertise. In contrast, optical genome mapping does not require cell culture and relies on automated bioinformatic analysis. For these reasons, we decided to implement OGM in our laboratory. We acquired the Sapphire system in May 2023, followed by installation, training, and familiarization with the assay over several weeks. We then compared optical genome mapping with standard of care techniques and completed the validation phase, which I will present today. We have been using optical genome mapping on a weekly basis since September 2024, and more recently, in October 2025, we installed the Stratus system, and its validation is currently ongoing. During these 2 years, we analyzed more than 300 samples of blood, bone marrow aspirates, amniotic fluid, and chorionic villi cells. These samples covered various indications such as infertility, hereditary cardiac diseases, developmental disorders, as well as hematological malignancies. For all cases, OGM was performed in parallel with standard techniques, including karyotype, FISH, chromosomal microarray, and long-read sequencing. So here is our wet lab workflow. For EDTA blood samples, after receiving the blood, we prepare 4 or 5 vials that are stored at -80. For bone marrow aspirates, we count the cells and pellet approximately 1.5 million cells in several vials and also stored at -80. For prenatal samples, such as amniotic fluid or chorionic villi, we maintain the cells in culture until reaching confluence, then count and pellet 1.5 million cells before freezing. So DNA extraction is performed on day 1 using frozen vials. The DNA is then stored at room temperature for 48 hours to allow homogenization. And on day 3, we quantify the DNA, perform labeling using Bionano labeling and stain kit, and proceed to scanning. After de novo or rare variant analysis, results are visualized and used as circus plots here in order to identify copy number variations or structural variations such as translocations and chromosomal deletions. For molecule qualities, we follow the recommended parameters for N50 that reflect molecule length, average label density, map rate, effective coverage, PLV, and NLV. So our weekly organization is as follows: DNA extraction on Tuesday, labeling and staining on Thursday, and scanning on Thursday and Friday, and data analysis over the weekend. With this workflow, we are able to process up to 12 samples per week. So as mentioned earlier, we performed a concordance study comparing optical genome results with standard of care cytogenetic techniques, including karyotype, FISH, and chromosomal microarray. We observed 100% concordance for all reportable findings detected by microarray, karyotype, and FISH. In addition, optical genome mapping allowed more precise identification of breakpoints, as we saw here in several translocations or inversions, compared with conventional karyotyping. However, we know that centromeric regions are poorly covered or masked in optical genome mapping. For this reason, balanced Robertsonian translocations or rearrangement involving centromeric regions cannot be detected by OGM. As illustrated here, translocation between chromosome 13 and 15 or between chromosome 22 and Y chromosome are not visible. Therefore, conventional karyotyping remains the only technique capable of detecting rearrangement within a heterochromatic region. So after the method comparison, we performed a reproducibility study using bone marrow and blood samples with known abnormalities, including translocations, trisomies, or monosomies. Analyses were performed in duplicate, intra-run, inter-run, and by different operators. All reportable copy number variation and structural variation were detected in precision study between runs. All molecule quality parameters were highly comparable between runs. We did not observe significant differences in N50, label density, map rate, or positive and negative variance rates. Overall, run quality was very good, with label density between 14 and 17 per 100 kb, and N50 above 230 kb. Following validation, we initiated a project called CartoCard, aiming to evaluate the performance of optical genome mapping in congenital heart disease. We analyzed both prenatal and postnatal samples obtained from amniotic fluid and blood samples. So first, we compared quality metrics between prenatal and postnatal samples. Our DNA molecule were longer than 200 kb. Effective coverage, map rate, and label density were within the recommended values. No significant difference were observed between the two groups, and we achieved 100% concordance with standard of care techniques, mainly chromosomal microarray and FISH. And in addition, OGM revealed additional findings in 3% of samples. I will now illustrate how optical genome mapping improves visualization of structural variants through several case examples. So the first case is a 26-year-old female with infertility and repeated implantation failure. Conventional G-banding analysis showed a seemingly normal karyotype. However, optical genome mapping identified a 30 megabase inversion in chromosome 5 between Q31.2 and Q33.1. When we re-examined the chromosome 5 on the karyotype, this inversion was extremely subtle. This illustrates how optical genome mapping can detect relevant abnormalities that karyotyping may miss. The second case is a 39-year-old male with recurrent deletions and duplications observed in his offspring. Several years ago, we identified a paracentric inversion in chromosome 1 between Q23.3 and Q25.3 detected by karyotype and FISH. With optical genome mapping, we identified 3 breakpoints on chromosome 1 located in 1Q21.3, 1Q24.3, and 1Q41. These findings demonstrated that this rearrangement was in fact an insertion of the Q21.3, Q24.3 segment into the 1Q41 region. These findings were confirmed also by long-read sequencing, which identified nearly identical breakpoints. The third case, is a 36-year-old male with infertility and an impaired embryonic development. A balanced reciprocal translocation between chromosome 1 and 7 was already known. Using optical genome mapping, we were able to precisely identify the genes involved at the breakpoints. The breakpoint on chromosome 1 disrupted the MSH4 gene, providing a clear explanation in the observed phenotype. In conclusion, optical genome mapping allows detection of genomic variations at much higher resolution than conventional cytogenetic techniques. It enables the resolution of complex chromosomal rearrangement, does not require cell culture, and eliminates inter-observer variability through automated bioinformatic analysis. Compared with chromosomal microarray analysis, OGM also provides additional structural information, such as distinguishing tandem duplications from inverted or mirror duplications. However, the technique requires technical expertise and molecular biology skills, and conventional karyotyping is still necessary for detecting rearrangement in heterochromatic regions. Our next step in the lab includes completing the validation of the Stratus system to increase throughput and implementing the VIA software in our laboratory. Finally, I would like to thank the entire cytogenetics and constitutional genetics lab at Amiens University Medical Center, and especially all colleagues involved in this study. And I want to thank also the Bionano Genomic teams for their outstanding and constant support. And thank you very much for your attention.